Hello everyone. So in this video we will be doing about the experiment which was done by Hersey and Chase in 1952 in order to prove that the genetic material is actually DNA. Okay. Now see, they worked with viruses, okay, that infect bacteria. Okay, these two scientists, they worked with viruses that infect bacteria. That means it is bacteriophage. Bacteriophage are nothing but they are viruses. It is the virus. This is not bacteria, it is a virus. Okay, virus that infect bacterial cell. Okay, bacteriophage. Now what did they do is they cultured some of this bacteriophage. Okay, some of the bacteriophage. Okay, some of the bacteriophage, that is half, let's say half of the bacteriophage and half of the bacteriophage, they cultured in radio they cultured in a medium which contained radioactive phosphorus. Okay, and some of the bacteriophage, it was cultured in a medium, it is a medium. Okay, culture medium. Half of the bacteriophage they cultured in a medium which contained radioactive sulfur. Okay, virus was cultured, this bacteriophage was cultured. Okay, now the viruses, the bacteriophage that was cultured in the medium which contained radioactive phosphorus, they contain this virus. Okay, this bacteriophage which was cultured in a medium which contained radioactive phosphorus, they have radioactive DNA because phosphorus is present in DNA, not in protein. Okay, phosphorus is present in DNA. That's why when some of the bacteriophage, it was cultured in a medium that contained radioactive phosphorus, the bacteriophage then have radioactive DNA, then contain radioactive DNA. And some of the bacteriophage, some of the virus which was cultured in a medium that contained radioactive sulfur, that bacteriophage contain radioactive proteins because sulfur is present in proteins but not in DNA. Okay, now remember one thing, this bacteriophage, okay, the virus that infect bacterial cell, they have only DNA and protein inside their cell. Bacteriophage that infect bacterial cell, virus that infect bacterial cell, they have biochemical DNA and protein only inside their, I would not say cell, inside their body. Okay, so now it is clear that some of the bacteriophage they contain radioactive DNA, some of the bacteriophage they contain radioactive proteins. Now what did they do after that? Now these bacteriophages, okay, these bacteriophages were allowed to infect bacterial cell. Okay, they infected E. coli cell, name of bacteria. Okay, they infected E. coli cell. Now when these viruses infect, suppose I will draw like this, okay, virus particles, you can see in the book also, this virus particle, okay, they were allowed to infect E. coli bacterial cell. Similarly here also this, okay, they are allowed to infect bacterial cell. Now they infected bacterial cell, okay, and whatever biochemical, whatever genetic material, whether it is DNA or protein, whatever it is, it got inside the bacterial cell, okay, it got inside the bacterial cell, okay, it got inside the bacterial cell. Now this coat, okay, virus coat, that means outer layer of the virus, okay, the coat of the virus was removed from this bacterial cell. Only whatever biochemical uh, was there inside this virus particle, it got inside the bacterial cell, and this coat, okay, outer coat of the virus, it was removed by centrifugation. It was removed by the method of centrifugation, okay. Only the biochemical, it got inside the bacterial cell, E. coli bacterial cell, okay. Now what happened is, when this genetic material, okay, the genetic material from the virus get inside the bacterial cell, the bacterial cell treats this virus biochemical, okay, as their own, okay, and make more virus particles, okay. They consider, this bacteria consider the foreign particles which came from the virus particle as their own, and it will multiply inside the E. coli bacterial cell. Now what happened is, this bacterial cell which was infected by virus that contain radioactive DNA, okay, and this bacterial cell which was infected by virus that contain radioactive protein, okay, at last this was radioactive. This bacterial cell which was infected by viruses that contain radioactive DNA was radioactive, okay, but this bacterial cell, E. coli bacterial cell, which was infected by viruses that contain radioactive protein, this bacterial cell was not radioactive. It was not radioactive, okay, but they contain protein, okay, virus particle is there, they contain DNA, protein from this virus particle. This E. coli bacterial cell, this cell also contain protein and DNA from this virus particle, okay, but the bacterial cell, E. coli bacterial cell, was radioactive, but this E. coli bacterial cell was non-radioactive. That means it is actually DNA. It is actually DNA that is acting as a genetic material, okay, making this E. coli radioactive, okay. There was also DNA, but this DNA was not radioactive because here radioactive was protein, okay, so the bacteria was not radioactive here, because protein is not a genetic material. Protein contain okay, this protein is radioactive, but it cannot transform this bacteria into radioactive, but This virus, which contain radioactive DNA, can transform this bacterial cell into radioactive form. So this way it proved that it was DNA actually which was genetic material, not a protein.